So I don't know where I left off, but yes, I'm going to be telling you a bit about the USGS Phycology Lab and some of the research we do here. The lab is located in the Caribbean Florida Water Science Center in Orlando, Florida. And the majority of our work is funded by the cyanotoxins and harmful algal blooms Integrated Science Team, or IST, of the Environmental Health Program. So the two primary activities of the lab here are to perform quantitative taxonomic analysis by microscopy, as well as cell isolation and culturing of algae. So here's just a slide showing some of the most common slide preparation methods that we use here. Uddermole is our go-to for phytoplankton quantitative counts. Sometimes, however, when we have an algal bloom, like in this sample here, this technique is not the most appropriate because a lot of cyanobacteria contain aerotopes, which is clusters of gas vesicles that allow these species to be really buoyant and control their buoyancy. So they won't settle in these uddermole chambers, and they kind of just get trapped up here with the cover slide. So when we have samples like this, I tend to use the Sedgwick rafter chamber. I mean, it's not the only thing I use it for, but this really helps when we have like a dense bloom because we're able to basically trap those floater cyanos under that cover slip. And then, you know, the Sedgwick kind of has its downside is that on the, you know, an upright microscope, you can only use a 20x objective. You can't go higher than that, and that's really not sufficient to do to make accurate identifications. So we can use it on the inverted scope. However, now you're looking, you know, your objective is looking through the bottom plate, which is a lot thicker, and you can adjust the aperture on your objectives to get a clear image. But like at least on my scope, the 60x, even adjusting the aperture on the objective, it's still things are a little blurry. So actually just playing around with like, oh, what can I do here? I realized like, you know, I could just flip these chambers upside down because once you have the sample in there, the cover slip makes a really tight seal with the chamber. So I tried just, you know, turning it, flipping it upside down and counting it like that. And it actually works wonderfully because now you're just looking through a cover slip and you get really good image resolution. And you're still getting floaters in here because this chamber has a depth of 1 millimeter. So then you just have to be sure that you're counting, you know, on the top plane where all your cyano floaters are going to be, but they're trapped there. And then do another count at on a lower plane, which is where basically everything else ends up being. We also do the semi-wet mount and dry mount method. I don't tend to use these for quantitative counts unless it's like for periphyton samples, but I do prepare these so I can take like really nice images on my 60 and 100x oil objectives, which I only have on the upright microscope. So this is a picture of our microscope room. We have the inverted microscope over here on the left, and that's an Olympus IX53. And then our upright microscope is a Olympus BX53 on the right here. And this is equipped with epifluorescence. And like I said, it's the only one we have oil objectives on also. So both of these microscopes are connected to a central computer with Olympus CellSense imaging software. And you can see a cool rotifer. I think that's from a Lake Tahoe sample there on the screen. Then the photo on the left is my reference library. I also listed some of the online website references that I use, including DONA, of course. But I know all you on this call are familiar with these sites. So here you can see how we use epifluorescence to distinguish between cyanobacteria and eukaryotic algae. So the photo on the left here is no filter using bright field. And we have microcystis and then some greens scattered throughout. And with the violet filter for chlorophyll A, you see both the cyanobacteria and the greens. And then when we switch over to the green filter for phycocyanin, you can see that the greens are no longer visible and you're just looking at your cyanobacteria. And this is just a screen grab of the CellSense imaging software that we use. It has tons of functions. It's really great, but I'm sure we're all familiar with one version of another of imaging software that we all use. So I'm not going to get into detail about that. And then, so the other primary lab activity is isolating and culturing algal cells. So we prepare and sterilize the culture media here. We use different recipes depending on, what we're trying to grow. Kind of need to play around and see what works best. But basically, we take a subsample of live material, place it onto a Petri dish, and scan it with the stereo microscope. And then we do a coarse cell picking from the Petri dish and transfer it into these 6-well plates using a Pasteur glass pipette, which basically you can melt the tip off and use like forceps to form the tip and narrow the tip so that we're, picking hopefully just the individual cells. But so once we have them in the 6-well plate with our culture media, we put them over here onto the inverted scope that's equipped with the Narashigi micromanipulator system. And here we do the serial washing in the 6-well plates. And it also uses a glass capillary that can be manipulated. Narashigi sells like a pooler system that has like a bunch of different settings that allows you to shape the tip of the capillary in different ways. So that's really cool. And then once we have unialgal cultures in these well plates, we transfer them into these 250 mil glass flasks with fresh media. And we maintain batch cultures here. So we have, a finite volume of media for the cells to grow. We don't add new media. Instead, we do routine serial transfer to fresh media in flasks approximately every 3 months, or depending on, the species, what they may need. And they're grown under ambient conditions. So they're on this shelf placed next to a northwest facing window. So we don't control light or temperature or anything. They're just growing in natural conditions and it works quite well. So I'm going to just go over some of the current projects we have going on in the lab. I'll start off with our Lake Okeechobee phytoplankton and HABs ecology study. So I think I missed a slide, but so Lake Okeechobee, as many of you may know, is located in Central Florida. It's the 8th largest freshwater lake in the US, and it's really important part of South Florida's water supply and flood control systems, and is also a popular recreational spot. The lake is surrounded by agriculture and urban areas that unfortunately have contributed to nutrient over-enrichment in recent decades, and that's largely been blamed for the increased incidence and intensity of HABs in the lake. Additionally, the hydrology of the watershed has been greatly modified from its historic flows. And is very heavily managed, but there are restoration projects underway to restore some of the natural flow conditions to restore some of the natural flow conditions. which were, water coming from the Kissimmee into Lake Okeechobee and then flowing down to the Southern Everglades. But that got channelized and redirected to urbanize South Florida. So now we have large amounts of water being pushed out the Caloosahatchee River to the west and into the Gulf of Mexico, and then through the St. Lucie River to the east and into the Atlantic. And this connectivity is thought to influence HABs in the rivers and along the eastern and western coasts of Florida as well. So we were funded by the Army Corps of Engineers to do this in-depth study on the ecology of Lako phytoplankton and HABs. And they funded us for 3 years, during which time the field work was being conducted, but the ongoing sample and data analysis and publications are being supported by the Environmental Health IST. I mean, we collected so much data as part of this project. There was no way to finish everything in those 3 years, but so the objective of the study was to understand the patterns and drivers of spatiotemporal distribution of phytoplankton assemblages and their potential and actual cyanotoxin production, right? So really focusing on the cyanobacteria that we know are the dominant bloom formers in this lake. The approach was to use a combination of monthly field surveys over 32 months, and also temporally replicated in situ nutrient amendment mesocosms that were conducted on the lower Caloosahatchee River down here. And for both the field and mesocosm studies, we looked at complete phytoplankton quantitative taxonomic analysis. This, however, was done using a flow cytobot. We actually contracted this work out to Phycotech because there were just like so many samples being collected for this study that we needed, a quick turnaround and a rapid measurement method for the IDs. We also measured cyanotoxin biosynthesis gene concentrations for common toxins, which include saxitoxin, microcystin, cylindrospermopsin, anatoxin, and nodularian, and then also measured the actual cyanotoxin concentrations. However, the cyanotoxin data is going to be coming out in a future paper because it's kind of been taking a long time to get all that data in. So there's not going to be a whole lot on that here. But the phytoplankton assemblage dynamics was published in Harmful Algae. It's still kind of in a preprint form, but should be out in the next issue. And so what we found was that Lake Okeechobee phytoplankton assemblages are dominated by cyanobacteria, and they are strongly spatially structured. And they form these 7 significantly distinct clusters or lake zones shown here on the map. The graph down here is showing the total algal cell densities at each of those zones averaged over the 32-month sampling period. And we could see that the highest total algal concentrations were found in the Northwest Lake Zone, so this area up here, and decreasing as we move south in the lake. And these patterns are thought to be connected to the flow patterns of the lake, where the majority of freshwater inputs are coming from the Kissimmee River, as well as other canals and creeks flowing into this Northwest Lake Zone. And then the greatest outflows are happening out the Caloosahatchee River and through the St. Lucie, but to a lesser extent. And this is so a list of the cyanobacteria we identified, several of which are known toxin producers, noted with the red next to them, showing what toxins they're known to produce. And then the graph here is just showing the total cyanobacteria cell densities partitioned by those different genera for each lake zone. And you can see there is considerable community dissimilarity among these lake zones in cyanobacteria assemblages, as well as, the complete phytoplankton assemblages. So The cyanobacteria that contributed the most to the total biomass were Aphanocapsa, Merismopedia, Planktolingbia, and to a lesser extent, Raphidiopsis and Microcystis. And the bottom graph here is showing the distribution of the biosynthesis genes from left to right, so microcystin, cylindrospermopsin, anatoxin, and saxitoxin. All 4 genes were present in all lake zones. And so the information we get from biosynthesis gene concentration, it represents the potential for toxin production, so that we know the genes are present, but we don't know if they're turned on. So we're still waiting on that ELISA data, measuring actual cyanotoxin concentrations to tell us if, the toxins encoded by these genes were actually were also present in the samples. So then we used a distance-based redundancy analysis on 16 abiotic variables to search for the strongest explanatory variables of those spatial patterns that I just showed, and also of temporal patterns. So the results from the spatial models showed that nitrate, phosphate, and alkalinity explain the greatest proportion of the total spatial variation of phytoplankton assemblages, and the most parsimonious model explained 93% of that spatial variation. So very well explained by these abiotic variables. And we saw, kind of surprisingly, but not really, that those top 3 explanatory variables were negatively correlated with the 3 different biomass metrics that we looked at, so chlorophyll, cell density, and biovolume. And even though it may seem counterintuitive, usually we're like, oh, there's high nutrients, we're going to have high abundance or high biomass. But this inverse relationship has been documented previously in Lake Okeechobee and other eutrophic lakes. And it's attributed to this lag between peak inorganic nutrient availability and then peak abundance during the phytoplankton boom and bust periods in systems that are they're chronically enriched with both N and P. So as phytoplankton reach carrying capacity during the boom phase, they're at their highest biomass, but they have depleted the nutrients in the water column. Then they reduce light availability by self-shading. That leads to declines in biomass as the bloom senesces and eventually dies off, in turn replenishing the nutrient pool as that matter decomposes, setting up for the next boom phase. So the temporal patterns of assemblage structure in the lake were also examined, but there was no evidence of succession across meteorological seasons or subtropical wet dry seasons. The temporal model only explained 48% of the total temporal variability, suggesting potential stochasticity and lake-wide shifts in assemblage structure over time. However, the strong spatial structuring of assemblages that we just showed may preclude a lake-wide succession patterns and the need to examine temporal variability individually within those lake zones. Also, there might be environmental variables that were not included in the models. These are all abiotic factors, so we don't have any biological variables, and that could also be playing a role in this variability that is unaccounted for by our models. So finally, we used Titan, which is threshold indicator taxon analysis. It's a package available in R to identify thresholds along the gradients of the environmental variables that were selected as the strongest explanatory variables in the spatial patterns, right? Because the temporal patterns weren't very strong. So I'm just showing the nitrate gradient here as an example. So the analysis tells us the species-specific change points, which is the value along the gradient where the greatest change in abundance occurs for each of these indicator taxa. And then the higher the z-score, so the darker the color, is the greater the indicator power of the taxa. And then Titan also calculates a community level threshold, which is the value where the assemblage cumulatively shifts in a significant way. For nitrate, that was 182 micrograms per liter, and there are also confidence intervals associated with these thresholds, right? So that community level threshold was used as a cutoff to characterize taxa that are associated with bloom and non-bloom conditions. So the second part was the mesocosms. Here we were testing the effects of different nutrient species different nutrient species, including nitrate, ammonium, urea, and phosphate on phytoplankton structure, heb formation, and toxin production. We conducted a total of 6 independent experiments from 2020 to 2021, but I'm only gonna present the results from the first 3 where the treatments were just nitrate, ammonium, and phosphate. So urea was a treatment in the last 2 mesocosms. And so these were conducted in the summer, fall, and winter to understand how these responses to nutrient additions might differ during different times of the year. So the experiments were run for 72 hours. We collected a baseline sample before the dosing on the first day. And then samples were collected before dosing on previous days. So T24 and T48, and then T72, we just collected a sample on the last day, but obviously there was no dosing then. We analyzed the samples for phytoplankton composition, cyanotoxin, and cyanotoxin biosynthesis gene concentrations. So for the nutrients, we measured them before dosing, and we also took measurements 30 minutes after dosing so we could determine the effect of the dosing solutions, right? So what we saw is that in all 3 deployments, the added nutrients were drawn out of, significantly drawn down from the water column within 24 hours of dosing. And that effect was, really more obvious in the June 2020 deployment. So then we looked at different biomass metrics, and we saw that in June, cells per microliter here. So cell concentration, chlorophyll, and phycocyanin, which is the cyanobacteria specific pigment, were all significantly elevated with the nitrate and ammonium treatments. In September, we saw no effect of any treatment. And then in February, we saw ammonium result in elevated chlorophyll and phycocyanin, but not total cells. I mean, they were higher in the NH4 treatment, but it wasn't significant. So this is the assemblage responses just for the June 2020 experiment where we saw the greatest effects. And here we can see that cyanos, green algae, and diatoms were the dominant phytoplankton in June. And then the dominant cyanobacteria, similar to Lake Okeechobee, that I showed you before, was Aphanicapsa, Merismopedia, and Planktolingbia. Interestingly, Microcystis, the dominant bloom former in the Lake Okeechobee system, maintained consistently low abundances and didn't respond to any of the nutrient additions. So we were unable to elicit a bloom of any cyano, but we expected Microcystis with our nutrient additions. So we didn't see any significant shifts in assemblage structure in response to the treatments. So that the taxa that were already dominant at the beginning were the main contributors to that increased total biomass that we saw with the nitrate and the ammonium treatments. And we also measured the net rate of population increase for the 4 treatments, which further confirm that no significant shifts in assemblage structure occurred. Okay, so that is it for the Lake Okeechobee. We're gonna move on to some of the other projects, and these are one-sliders. So we have a project in Tampa Bay, and Tampa Bay and the Gulf of Mexico, as you may know, experience frequent HABs of Karenia brevis, known as red tides. Karenia brevis produces a potent toxin called brevetoxin, which is harmful to human and wildlife. Pyridinium bahamense is another dinoflagellate that is a common HAB former in Tampa Bay and the Gulf of Mexico, and it produces saxitoxins, which is the cause of paralytic shellfish poisoning. But it also produces bioluminescence, which can result in beautiful nighttime displays in this area. Then pseudonychia, which produces domoic acid, the cause of amnesiac shellfish poisoning, is another common HAB former in this area. So I'm working with folks in the USGS St. Petersburg Coastal and Marine Science Center, who are experts in ocean chemistry, on a study to understand if there's a link between ocean acidification and HABs in Tampa Bay. So we conducted this preliminary sampling in July and August of 2023 to look at phytoplankton distribution along a pH and salinity gradient. So that's increasing salinity and pH down this way. And shown here are some of the taxa that were identified during the July sampling. Rhizoselenia, Thalassionema, Chaetoceros, pseudonychia were dominant in these samples, and dinoflagellates were diverse, but interestingly, we didn't observe any Karenia brevis, but there wasn't a red tide during this collection. Still, it's kind of surprising we didn't see at least like background Karenia during these samples. So another project, this was a preliminary investigation of algal assemblages and water quality and HABs in San Juan, Puerto Rico. So this was a collaboration with the Puerto Rico Department of Natural and Environmental Resources. So we were looking at just characterizing the algal assemblages and the status of HABs in Caño Martín Peña, which is a channel that connects the San Juan Bay and the San Jose Lagoon, which are all located in the San Juan Bay National Estuary, which is one of the 28 estuaries belonging to the National Estuary Program. The mission of that program is to protect and restore the ecological integrity of estuaries of national importance. So this estuary not only carries immense ecological value, but it's also really vital to the economic development and food security of Puerto Rico. So the CMP, the Caño Martín Peña, is a channel, like I said, that connects the bay and the lagoon. It runs through the heart of San Juan, and it's one of 20 designated urban water sites under the Urban Water Federal Partnership, through which the pilot study was funded. And the partnership aims to revitalize urban waters like the CMP that have become polluted and degraded, with the goal of protecting ecologically valuable ecosystems and improving conditions to the surrounding communities, which are often underserved and economically distressed. So yeah, although you know, algal studies in this area are extremely limited, and those that exist haven't been published, harmful algal blooms have been reported as part of the degradation of the CMP. Like we were out there, the captain of the boat we chartered, he does fishing charters mostly, but he also like helps PR a DNER with their sample collection, and he was telling me, you know, he sees harmful algal blooms out there, and it's mostly in the wintertime. So our samples were done in the summer, and we didn't see a bloom at any of the sites. So definitely want to go back in the winter. But yeah, I'm still working up the data on this. So just some pretty pictures of us in the field, in the lab. And we did also collect some samples on land because there's a lot of flooding that happens in the surrounding communities of the channel, because the channel has become so just like polluted, but with like really large items. So there's not really a whole lot of flow through the channel. And then, we get this overflow into those communities. Another study is a look at the HABs and toxins in several different fish and wildlife refuges. So our IST, the EHIST that I work with, is working with the US Fish and Wildlife Service to assess HABs and toxins in a selection of wildlife refuges where HABs have been reported. The sampling was conducted at 14 different refuges shown here on the map in August to September of 2023. Some of these refuges have 2 locations within them, so it was a total of 22 sites. And the ones that are in red are where we had HABs during collection. And then the photos, the arrows pointing to the photos, are indicating the dominant taxa, the bloom forming taxa, and they were cyanobacteria. As you can see, for all of these, they were all cyanobacterial HABs. And then last but not least is our Lake Tahoe study. So again, our IST is doing an algal and toxins assessment in Lake Tahoe. This normally pristine lake has recently experienced some nearshore algal blooms and signs of water quality degradation, especially down here in this area. And so our IST received a congressional mandate in 2023 for Lake Tahoe monitoring. Sampling started the summer of this year. And here are some photos of our recon trip in March, where we were developing the sampling and study design along with the folks at the Nevada Water Science Center. And so we came up with 3 transects in the southern lake where the algal blooms have been reported. And these at each point along the transect, we're also sampling vertically. So really get the whole 3D picture of what's happening there. But then we also included these other individual sites around the rest of the lake. So one on the west, one in the north, and another one on the east. And I'm just starting the algal counts on this now, so I don't have any data or pictures for you guys on this yet, but stay tuned. And that is the end of my presentation. There's my contact information at the bottom. And yeah, I'll take any questions now.
All right. Hey, thanks, Vivi, for the presentation. Interesting to see how far you've spread the influence of this lab in terms of some of the other places you're working beyond just Florida.
Yes.
If folks want to put comments in the chat or questions in the chat, you're more than welcome to do that. Otherwise, we don't have we've got about, looks like we got 23, 24 people on the line. If you just want to use the hand raise thing, you feel free to ask a question. I see Evelyn, you've raised your hand. Go ahead.
Hi there.
Hey.
I don't know if I'm showing. I'm walking my dog, and it's like freezing out.
I know. I see your giant coat.
Anyway, great presentation. It is such comprehensive work, especially the Lake Okeechobee combination of the surveying that has gone on for so long, and then the experiments as well. I was just curious about, you're showing quite a bit of nitrate regulation. And I just wondered if you've looked at like the N to P ratio in these areas in the lake to see, what that looks like, and whether the algae are kind of reacting to changes around that sort of red field or something like that.
Yeah, so that's actually something I get into in the journal that we're trying to,
Okay.
it got invited to a special issue, Lake O for lake and reservoir management. So I didn't get into it here, but it's definitely something I discuss because the concentrations of both nitrate and phosphate in the river were above like the Reynolds proposed saturation thresholds, which is like 110 to 130 for nitrate and like 10 for phosphate. So the background concentrations are already like super saturating, but we're not
Cool. Yeah.
we're not constantly seeing blooms happening in the river, and we
Right.
think that's because the river has a lot of dissolved organic matter. It's a very colored river.
Oh, okay.
Yeah, so that's likely mitigating because you saw how
Sure.
much nutrients we added in there, and that wasn't even
Yeah.
enough to get it to bloom.
Right. Okay.
So yeah, so we're thinking at that point, at those saturating levels, the ratio really is not going to have an effect, right?
Sure. Sure.
Because
Sure.
yeah,
Yeah. The light
um,
limitation set to. Cool.
yeah. But we
Thanks.
do provide the ratio data in the paper, so.
Okay. Cool. Great.
Are you at a stage with that paper that you can share it with folks, or is it still
The
in
mesocosm paper.
the harmful algae paper, the drivers
Oh,
paper?
yeah, it's up. So it got Bureau. Yeah, we have a very strenuous review process for journal articles. But that one does have BAO approval, which is the final approval, and it's a preprint. The preprint, harmful algae puts the preprint up for the public.
Okay.
I would just suggest you wait till they finish my proof corrections and they publish it in this next issue, because there were a couple of typos we caught in the proof, so I'll just wait for that.
Cool. Again, folks, if you'd like to either type a question in the chat or just use the reaction button on the bottom to raise your hand, we can easily take any questions you have. I've got a couple. I was just curious. You said you were doing you're doing a bunch of culturing as part of this. What's sort of the purpose of the culturing that you're doing? Is it
Yeah, so I knew I was already like over on my slide, so I didn't include get into the culturing, but this is another like extra slide I had of the workflow, right? And so for the culturing part of things, what we're using those cultures for, one of the work plan objectives for our IST group is a polyphasic analysis of cyanobacteria that basically is attempting to harmonize morphological and molecular based cyanobacteria identifications and descriptions. So the culturing helps us with that because, we're able to get these unialgal cultures sequenced and then, have the, pretty flora vouchers and morphological descriptions that can be published together with the molecular data. So a lot of the sequences for cyanobacteria, they're not the best. You'll have like sequences and it'll barely have any information linked to it, like not even, where it was collected if you're lucky. So our idea was to have a database that had like the sequence data, and then, you had all the ecological, the odd ecological data, as well as the morphological descriptions, all kind of in one database. So that's one thing. Another thing is testing toxin production of those isolates. And so the culturing kind of fits into all these studies. So if I get live samples from any of those studies, I'm like looking and seeing what I can culture. So I have like cultures from a wide range of places. So this is kind of happening in conjunction with all these different studies, but testing toxin production of isolates is another one. Also, using them in experimental studies. So I'll have people reach out to me and be like, hey, we're running this experiment. Can we get some of your cultures to do that? I'm not running any experiments with them right now, but we have them if we want to design something like that. And then they also serve as living flora vouchers for some of these studies.
Are you depositing them in any of the other algal culture collections?
No, not yet,
No.
because
Okay.
unfortunately I'm like running this lab by myself, and the culturing is yeah, I need to hire staff like really bad, and culturing is kind of falling to the wayside. So they're not in any condition to be deposited. But once, once we get that program back up and running and the way it needs to be, then yes, definitely we plan to do that.
Cool. Yeah, I wonder if cyanobacteria can be more easily like frozen and just stored as as rather than having to continually transform. I
Yeah.
know diatoms are problematic in that way, but I wonder if cyanos could do it. So
No, cryopreservation for cyanos is pretty common.
good.
So that's probably something I need to start looking into because I can't keep doing like the serial batch culturing transfers. It's a lot of work. So yeah.
And Matt from UTEX has weighed in. Thank you, Matt. We got a question in the chat. Which Rhizoselenia species do you consider harmful, and do you have any ecological parameters for it? That's coming from Layla.
Oh, I don't know. I don't know that Rhizoselenia is considered a harmful species. It's just one of the dominant taxa that we found in that study, but
Okay.
there's no indication that it produces any toxins or is even really bloom former. But it's just a very common taxa here.
What was the taxon that was producing the saxitoxin in that Tampa Bay project?
Oh, so that's Pyrodinium bahamensi
Oh,
produces saxitoxin.
okay.
And then we have the Crania brevis, the other that's the one that is really causing the majority of the blooms, like the red tides. And then I did find the pseudonychia, not not at bloom levels, but it was present in these samples, and that one also produces a pretty nasty toxin, domoic acid, that causes.
Were you picking up domoic in your samples?
So I don't have the toxin data back yet.
Oh, okay.
Yeah,
Got
so
it.
we're still waiting to put all the pieces together, the chemistry, the taxonomy, and the toxins.
Got it. Sarah's got a question.
Yeah, I hope my internet connection works. Vivi, thanks for a really interesting talk. And I'm curious if you're working with any of the USGS remote sensing group to match these compositional data with satellite signals.
So yes, so our IST is involved. I don't know if you've heard of the Cyan website. I'll probably look it up. But I did some ground truthing counts for that program, but Cyan is a web application, also a phone application. You can download it on your phone. And USGS was working together with NOAA and NASA and some other folks to create that application. But really, that's the extent of my involvement with that. I was recently at the Denver Science Center. Oh my God, what's it called? I was at a conference. Sarah, you know what I'm
Colorado
talking
Water
about?
Science Center.
No, not the big
Federal Center.
The Federal Center, yes. And so we took a tour of their remote sensing lab and all the capabilities they have. And I really want to reach out to those folks and start up collaborations to do some integration of remote sensing with the field sampling, even using things like drones to kind of do a scouting of blooms, but yeah, USG.
I can get you in touch. Tyler King is someone who's doing some really amazing work with remote sensing and remote identification, who's working across the country, and I'll forward some things to you.
Oh, perfect. Yeah, I'm super interested I got like a GIS certificate during my master's program, so I'm like, I need to put that thing to use. I really like remote sensing.
All right, thanks, Sarah.
Awesome.
I've got let me see if we got any other questions popping up in the chat. We haven't seen any other questions in the chat, but I just wanted to follow up. So you're talking about Lake Okeechobee or Okeechobee. And I'm up here in Minnesota where we're almost got ice on all the lakes now. And you brought up a comment about Okeechobee and sort of your bloom sequencing. And it sounds like it's almost, I guess what I'm questioning is how much seasonality do you get to your blooms? Or is it just this bloom and crash, bloom and crash, bloom and crash thing that goes on in these Florida subtropical big lakes?
So yeah, so the seasonality, apparently I marked all this up accidentally. So the seasonality in Lake Okeechobee, it wasn't as predictable as the spatial patterns, right? So there is a whole lot of variability we're seeing here, but we weren't able to attribute that to any like any successional shifts between, say, dry and wet season that was happening, on an annual basis, like a predictable cyclical or seasonal way. And I was saying that could be for Lake Okeechobee because it is so big and because we did see that strong spatial structuring, so our study was the first to kind of say, hey, there are these distinct zones of phytoplankton assemblages, but previous studies have shown zones pretty similar to those of distinct water quality. So we're thinking that those zones might be experiencing their own temporal patterns and that we probably need to be looking at those individually rather than lake-wide shifts over time because it's so heterogeneous.
Interesting. Last question I had on my list was just how your toxin analysis that you're doing, how is that done? How are you guys doing? Is ELISA, HPLC? What's the strat down there?
So we do ELISA as like the quickest one, and I'm not doing the toxin analysis myself here. Those are done at the Kansas Water Science Center by Keith Lofton's lab. They're like the chemistry folks. But they also use mass spectrometry, right? So that's it. Yeah, it's going to be a lot more accurate, but it's also a lot more expensive and time consuming. But we are going to have for the Lake Okeechobee stuff, which is why it's taken so long. But we have a lot of the ELISA data back, but it comes in like in random order and it's not complete. So we're just waiting for everything to be finalized, but we're going to have ELISA data, we'll have the mass spectrometry data, and then we're also going to have like some proteomics data. Something I didn't mention is that I should have mentioned actually. So this study was done in collaboration with folks over at Nova Southeastern University and the Florida Gulf Coast University, and they were focused on a molecular characterization of the cyanobacteria and other heterotrophic bacteria in the lake. So the same samples we collected and everything, we had the morphological analysis done, and then we had metagenomic studies and transcriptomic studies done. So there we can also really see if we had these cyanotoxin biosynthesis genes. And then the proteomics we're going to do is seeing, what metabolic processes maybe are happening. So there's a lot more to get into. This is like only scratching the surface with looking at just like the phytoplankton assemblages and what they're doing and what's driving their distributions.
Okay, thanks. In the chat, folks have been popping a number of links to various articles and things that feel free to click on those, folks. They won't be preserved in the recorded version, so click on them now while you can grab that link. There was a final question in there. Oh, we got a couple of final questions, but
Oh, I'm still sharing.
Anna wanted to know if there if you see variability strongly connected to precipitation, I assume in Okeechobee.
So precipitation did come out as one of the variables in the conditional test for temporal variability. However, if you look at it individually, it explained, less than 1%. Yeah. Or I'm sorry. It explains 7.6% of the temporal variability in seasonality. So no, yeah, not as much as we thought. And we included wind direction, wind speed, total solar radiation, precipitation, and temperature, and they really didn't explain a whole lot of the temporal patterns. So yeah, there's something going on there. It's either stochasticity or maybe there are biological controls which weren't included in these models that are driving the seasonal variability. Yeah. And then for spatial precipitation, wasn't a strong explanatory variable either. So yeah, that was kind of surprising.
Yeah, I would have thought maybe the way it was positioned with that stuff up in the north end of the lake, that might be some runoff situations that you were getting, flushing of nutrients into the system that were triggering stuff. But interesting.
Yeah, and that's the hypothesis. I mean, not just so much from precipitation, but also like water control schedules, water release schedules and stuff like that. But obviously that's linked to precipitation. So I'm not sure what's happening there.
Okay. Hey, Sarah, do you want to wrap up with your final question and then a little shout about DONA one more time, and then we can close things out here?
Thanks. Sure. Thanks as long as you can hear me okay. Okay. Yeah, Vivi, did you also look at cell density in terms of cell volume per, like cell volume per liter?
Yes. Yeah. So we looked at both cell concentration and biovolume concentration, or biovolume, and chlorophyll. So we had those three metrics of abundance.
And how much does that change things to look at volume? In terms of the past patterns over time and their relationships?
We didn't see, so I didn't run a model on biovolume, but it could be interesting to do that. But if I just looked at the, just a graph, like the ones I showed for cell density, you do see differences in like the relative proportion of the different taxa, obviously according to their size, but there's not a clear seasonality that shows up even looking at biovolume.
Okay. Right. Okay. Great. Well, thank you. I would just like to give a pitch here. I want to give a huge thank you to everyone that's contributed to diatoms.org. We started our fundraising just a couple of weeks ago, and we're 40% of the way there to our goal of being able to keep the servers running for 5 years. 5 years. So diatoms.org is free for everyone. So help us keep it free of advertising, because we don't want to go there at all. So every little bit helps. We have a lot of people who use this resource, and thanks so much.
Awesome. Thank you, Sarah. And great. That's great. That's so great to hear. Well, thank you, Vivi. It was wonderful to hear your presentation, hear all the great work that's going on down in Florida and beyond, and really appreciate it. We'll have the recording for this Diatom Web Academy posted on YouTube in the next week or week and a half, probably. And looking ahead, we right now don't have any additional Diatom Web Academies scheduled yet in the spring. Don't make us choose you. You feel free to reach out and if you'd like to address a global audience about some of the work that you're doing in algae or diatoms, please reach out and we'll get you set up. We are entering the holiday season here, so taking a little bit of a break for it for the next few weeks. Any last announcements to make, folks?
Oh, I just wanted to say I put the Cyan web app link in the chat for those that are interested in tracking blooms with satellites. Yeah, it's a cool app and you can get it on your phone too. So you want to find out if there's a bloom in your area, just go to the app.
All right. Well, thanks again, Vivi. Wonderful
You're welcome.
presentation and
Thank
great to
you.
see everyone. Everyone be well, okay? Well, bye.
Bye.
Thank you, Vivi.